All right, this is the second half of the Theory of Evolution lecture. This will correlate with Chapter 2 and Chapter 4 in the Explorations or the Essentials textbook. So where we left off from the last PowerPoint presentation, we were going over the various fields that influenced Darwin in developing his theory of natural selection, of evolution via natural selection. So the next field we're going to look at is the field of taxonomy and systematics. Before Darwin, most scientists did realize the need for classification of life forms or taxonomy. Early taxonomy was based on the idea that species did not change. Even Linnaeus himself believed that species are relatively constant. So again, remember, this was also influenced by religious doctrine of the time that assumed that a creator or a designer created species in their present form. So Linnaeus created a hierarchical system of nomenclature. So that word nomenclature is just a fancy way of saying naming, that provided unique names for all varieties of plants and animals. So every living species has a genus and a species name. For example, humans are Homo sapiens. Linnaeus developed a system of binomial nomenclature that is still used today. Each plant and animal has its own unique genus and species name. Linnaeus's book on taxonomy went through about 10 revisions, and with each revision he added more levels and more detail to the system. At the time, including humans in this scheme of binomial nomenclature was considered somewhat controversial because humans were considered to be separate from the rest of the animal kingdom and created in God's image. So this is, don't feel like you need to memorize this slide here. This is just to give you guys an example of what the system looks like. So at the highest end, we're looking at kingdom, and then subkingdom, phylum, subphylum, superclass, class, subclass, order, suborder, superfamily, genus, species, and subspecies. So for example, we share an order with all of the other primates. We share a superfamily with the other apes, gorillas, orangutans, chimpanzees, and bonobos. We share a genus with other members like Homo habilis, Homo erectus, Homo neanderthalensis, Homo heidelbergensis. But again, don't feel like you need to memorize this chart, but essentially we'll be going from order and down, looking at the primate order. All right, the other field that influenced Darwin in developing his theory of evolution via natural selection, this is Thomas Malthus, and he's considered to be the father of demography. So another way to think of demography is population genetics. He came up with this idea that population size will obviously be limited by available food resources, and he wrote an essay on the principle of population and made the case that population growth will be limited by food supply. So this basic idea did influence Darwin in his idea of survival of the fittest. In any given habitat, there will be what we call a carrying capacity. So that means that the population cannot exceed a certain size without there being a degree of competition that means that not all individuals will survive. So in this graph here that you see on the screen, we've got the resources are relatively constant here, and then you've got the population size. So you see it begins to grow here. And then this here, the point of crisis, that's where that population size is exceeding the carrying capacity. So when the population size is beyond the carrying capacity, that means that individuals are starting to die out due to lack of resources. So Darwin applied this idea to his own of survival of the fittest or natural selection. All right, so the last field that we're going to look at that influenced Darwin in developing his theory was biology. So by the late 1700s, scientists began to argue for biological change in organisms. Lamarck speculated that animals change for self-improvement, and he actually coined the term biology. So Lamarck's theory of inheritance of acquired characteristics looks at the idea that animals will change anatomically in response to new demands or needs, and then will then pass those changes or those adaptations off to their offspring. So he emphasized the importance of interactions between organisms and the environment. So Lamarck got it partially correct. What he got wrong was the time frame. So I'm sure you may have in past biology classes seen this famous image of giraffes that are extending their neck to reach leaves on higher branches. So Lamarck made the assumption that this could happen in one generation time, when in fact any sort of biological change takes usually dozens, if not hundreds of generations. So his basic idea was right. He just got the time frame wrong. All right. So this film here, The Genius of Charles Darwin, this one is available in your module. So if you click on this film in your module, you can watch this film for extra credit. I also attach some film notes as well that you can use to guide you through the film, and I will collect those at the end of the unit for extra credit. All right, so the next few slides, we're going to talk about the basics of Mendelian genetics. We actually go into this in a little bit more detail in chapter 3 and 4, but it starts to introduce it here in this chapter. So in Darwin's time, he didn't necessarily understand the mechanism of how genetic material was passed from one generation to the next. So Mendel kind of added the other part of the equation that made it understandable how these genetic traits could be passed along. So Mendel studied pea plants. In his observation of the plants, Mendel concluded that a discrete physical unit was responsible for the transmission of traits, and we now call that discrete unit a gene. Mendel also discovered that the traits in the pea plants did not blend, so they were either one or the other. So these are now what we call monohybrid traits. So for example, a plant would either be tall or short, the pea would either be white or purple in coloration, etc., etc. So we call alternate forms of these genes alleles. All right, so Gregor Mendel is considered to be the father of modern day genetics. Mendelian traits, or sometimes you're going to hear them called monohybrid traits, which means one gene, one protein. Mendelian traits are genetically simple traits that are determined by alleles at a single locus. So a locus is just the location of a gene on a chromosome. Humans have 46 individual chromosomes or 23 pairs. The principle of segregation is stating that alleles will separate from one another during the formation of gametes. A gamete is a sex cell. So we'll talk more about meiosis in chapter 3. Meiosis is how sex cells are formed. But for now, just know that the principle of segregation is stating that these alleles or alternate genes are separating from each other randomly during the formation of gametes. Each human has the capability to produce over 1 million different unique gametes. The principle of independent assortment is stating that the presence of alleles of one trait will not necessarily affect the expression of the other trait. So for example, your skin color isn't necessarily affected by your hair color. That's just a random example. All right, so some vocab words to get out of the way. So a gene is the segment of a chromosome's DNA that is coding for a specific protein. So your chromosomes are essentially its nuclear DNA that's coiled up around proteins that we call histones. And our genes, our genetic code, are located within those chromosomes. So the larger the chromosome, the more genetic material you're looking at. So an allele would be an alternative or alternate form of a gene. So for example, there's alleles that code for blue eyes, brown eyes, green eyes. There's alleles that code for whether or not you can roll your tongue. There's genes that code for your earlobe form, whether or not you have a widow's peak, etc., etc. This term homozygous, so if you break that word, the beginning part of it means same. Zygous means zygote. So a zygote is a fertilized egg cell. So all of us, at least genetically speaking, are quite literally 50% mom, 50% dad. We get 23 of our chromosomes from the sperm cell and 23 of the chromosomes from the egg cell. So for example, if you inherit a dominant allele from both mom and dad, then at that locus, at that location, you will be homozygous dominant. So remember, you can't just say homozygous. When we're talking about genotype, you need to say the full thing. So homozygous dominant is 2 dominant alleles. And we're always going to use uppercase letters to represent dominant alleles. So in this example here, large A is a homozygous dominant genotype. And then the other type would be homozygous recessive. So homozygous recessive just means that you inherited a recessive trait from the egg and from the sperm. So that will be represented by 2 lowercase letters. So the term heterozygous, these are chemically alternative forms of a gene. So if you break that word up, hetero means different, zygous means zygote. So that simply means that you inherited a dominant allele from one parent, either mom or dad, and a recessive allele from the other. So if the genotype is heterozygous, you always write it as one uppercase letter and one lowercase letter, and the uppercase letter will come first. So it would be an uppercase A followed by a lowercase a. So in the heterozygous genotype, at least if we're talking about simple monohybrid traits, the dominant allele will actually mask the expression of the recessive allele in the phenotype. So that'll make more sense once we do some Punnett squares. So hold that thought. All right, so a few more vocab words. The term genotype. A genotype is the genetic makeup of an individual. So that's when we're asking about homozygous dominant, homozygous recessive, or heterozygous. So in this example here, we're looking at earlobe form. So in humans, there's two different forms of earlobes. There's free-hanging earlobes. That's the dominant trait. So if you have the dominant genotype, the dominant phenotype, then you're either homozygous dominant, big E, or you're heterozygous. So the phenotype, that's the physical expression. So that's essentially what that human looks like for that trait. So we're only going to be looking at one trait at a time, just earlobes or just tongue rolling or just the presence of a widow's peak, something like that. So in this case, we're just looking at earlobe form. So if the human has the dominant phenotype, the unattached or free-hanging earlobes, then they have two possibilities. They can either be homozygous dominant or they could be heterozygous. Because in the heterozygous form, that big E is going to mask the expression of the little e. The homozygous recessive genotype is only present in those humans that have the recessive phenotype. So that's the the attached earlobes. All right, so let's do a Punnett square here. So in this example, both mom and dad are heterozygous. So we need one uppercase E and one lowercase e. So when you're making a Punnett square, just like it sounds, you're going to draw a square, and then you're going to divide it into quadrants. So one line down the middle here, one line down the middle here. And it doesn't matter whether you put the male parent on top or along the side. You just put one parent on the top and then one along the side. So we've got the heterozygous dad and then heterozygous mom here. So in this first little box, you're going to take one from here, one from here. So you've got a homozygous dominant here. And then in this little box, you're going to take one from here, one from here. So one big E, one little e. So you've got a heterozygous child here. And then in this box, you're going to take one from here and one from here. So you've got another heterozygous child here. And then in this last box, one from here, two little e's, homozygous recessive. So this part is important when you're setting up a genotypic ratio. It's always going to be in this order, always homozygous dominant versus heterozygous versus homozygous recessive. So once you set it up that way, all you have to do is count. So each of these four little boxes, they're representing 25% of generation 1. So if we wanted to know the genotypic ratio, the first thing we're looking for is homozygous dominant. So we have one individual that's homozygous dominant. Then we're going to look at heterozygous. So we look at our boxes, we count 1, 2. We've got 2 individuals that are heterozygous. And then homozygous recessive, we have one individual that's homozygous recessive. So our genotypic ratio is 1 to 2 to 1. The phenotypic ratio, so remember phenotype is the physical expression, so we're simply looking at the dominant genotype, the dominant versus the recessive. So remember for the phenotype, we're going to combine homozygous dominant with heterozygous. So we want to count 1, 2, 3. So as long as there's at least one big E, that means that that person's phenotype will be the dominant free-hanging unattached earlobes. So we have 3 individuals that will be the dominant and only one that will have the recessive attached earlobes. So genotypic ratio in this case is 1 to 2 to 1, and your phenotypic ratio is 3 to 1. All right, when we're talking about ABO blood typing, this is an example of what we call codominance, because both the allele that codes for blood type A and the allele that codes for blood type B are dominant. The allele that codes for blood type O is recessive. So each person will have one of 4 different blood types. So again, we're remembering that we're inheriting 50% of our genetic material from the egg cell and 50% of our genetic material from the sperm. So on one chromosome, each person will have either an A, B, or O allele. And then on the other chromosome, let's say this one's from dad, we have either the A, B, or O allele. And the combination will determine that person's blood type. So there are two different ways you could be blood type A. If you inherit an A allele from both mom and dad, that makes you blood type A. If you inherit an A allele from one parent and an O allele from the other, then you're still blood type A, because remember that A is dominant over the O. If you have type B blood, two different ways to get type B blood. If you inherit a B allele from both mom and dad, that means that you're type B. If you inherit a B allele from one parent and an O from the other, then you're still blood type B, because remember that B is dominant over the O. If you have AB blood type, this is the co-dominant condition. So what that means is that you inherited an A allele from one parent and a B from the other. So both of those alleles are going to be expressed in your phenotype and your blood type. So type AB blood, this is what we call the co-dominant condition, since both the A and the B allele are expressed in your phenotype. If you're type O blood, the only way this happens, you need to have a recessive O allele from both parents, from both mom and dad. All right, we're going to go ahead and pause here. I'm going to try to keep these PowerPoint presentations short and sweet for you, 15-minute segments, and then we'll continue on with this in the next presentation.